Thank you very much, Charlie. I want to give my personal thanks to both Charlie and Meredith for their outstanding work in bringing this together. I think this has, been a great addition to the field of evolutionary medicine, and I applaud their effort and their ongoing perseverance in making sure that this actually happens, especially in these complicated times. I'm also particularly excited about actually helping moderate today's session, where we bring in some of the rising stars in the field. We oftentimes get used to hearing from the sort of traditional leaders of programs and other things like this, but I think the real future for this field is in this collection of people that and the community that's growing, and the types of work that we're going to hear about today. So today we have actually at least 3 of the rising stars in the field, and so that I don't take too much of their time, I'll start by introducing our first speaker today. Our first speaker today is Caroline Amoroso. Caroline did her undergraduate work at Cornell and has a PhD in evolutionary anthropology from Duke, where she studied behavioral avoidance of fecally transmitted parasites in Lebers. She's currently a postdoctoral associate with Janice Anovics at the University of Virginia, where she's studying behavioral aspects of host pathogen interactions. Her talk today is evolution of host behavioral resistance to pathogens. And with that, I will turn the floor over, or the virtual podium over, to Caroline. So Caroline, take it away.
Okay, hi everybody. Thanks for joining us on what has turned out to be a pretty stressful day for some of us, at least in the United States. I am just gonna, can you see my screen? And can you see my slide?
Yep, looks good.
Okay, great. Okay, so I'm gonna start my timer. Thanks for being here. I really appreciate it. And I'm really excited to talk to you about the work I've been doing with Janice Antanovics on the evolution of behavioral strategies to resist pathogens. And we started this project about a year ago, before COVID, before social distancing was a part of our everyday lives. And we finished this paper many miles away from each other, completely remotely, while we were practicing the very kinds of behavioral resistance that we had modeled in this paper. So although I'm not going to be offering you any practical COVID advice today, this project has become surprisingly and unintendedly timely. So a central question in infectious disease research in fields like medicine, public health, and disease ecology is, why do some hosts get infected by pathogens and others do not? And there are many answers to this question. Hosts can vary in their genetic resistance. They can vary in their history of exposures contributing to their immunological resistance to pathogens. They can also vary in behaviors that expose them or protect them from pathogens. And so just as we can have genetic or immunological resistance to pathogens, these are the areas that evolutionary theory of resistance has traditionally focused on. We can also have behavioral resistance to pathogens. And we can see behavioral resistance to pathogens in some animal systems, like this well-known case of the Caribbean spiny lobster, which can detect the presence of a virus in conspecifics and choose to share a den with healthy lobsters rather than infectious ones. And these strategies have been called the behavioral immune system. This is a term that highlights the many analogies between behavioral and physiological forms of resistance to pathogens. But is this analogy simply a useful metaphor for talking about host avoidance behaviors, or are there actually evolutionary and ecological parallels between behavioral and physiological forms of resistance? So we took a theoretical approach to this question by considering 2 kinds of avoidance. One type of avoidance is what we call specific avoidance. And this would be similar to the disgust response in humans. We can detect when someone has symptoms of illness, or in the case of the lobsters, they actually detect cues of the virus itself and avoid contacting individuals who have cues of infection. Another type of avoidance is what we call general avoidance. And this would be a reduction of all social contacts to reduce the risk of contacting someone who's infected. And so this would be really helpful in the case in which the pathogen is undetectable or most of the infected hosts are asymptomatic, similar to what we're all practicing right now, social distancing. And then we asked the question, how does behavioral resistance evolve? We developed a theoretical model of a disease that's transmitted in a social context. We measured avoidance as the fraction of contacts that are not engaged in, and then we examined two forms of resistance that I just mentioned, specific and general avoidance. We also thought about some of the assumptions about what the costs of resistance are. So first, we examined a fixed cost of resistance. This is similar to how resistance costs have been modeled in physiological contexts. And then we also considered the case in which social interactions are themselves beneficial, and that the cost of resistance would be the loss of those beneficial interactions. And we wanted to compare our model outcomes to previous models of physiological resistance. And we deliberately kept this model as simple as possible by focusing on a single locus with two alleles so that individuals could either be a resistant or susceptible genotype. We had individuals interact in pairs. So these are the smallest, simplest groups, just with two individuals in them. And then we modeled two processes. First, we modeled the process of social interaction, so the formation of these groups of two. And that was nested within each generation of the model, where we modeled genetic change, the processes of birth, death, disease transmission, and density dependence in the model, in the population. And I'm going to throw some equations up. I don't want you to pay too much attention to the details of them. The point is to show you that even with all of these very simple assumptions about how this process works, suddenly things can become really complicated when we put them into these equations. And so even in this very simple case, it's very complex. So what I'm showing you here is how specific avoidance works in a population of only resistant individuals when the disease is present. So this is just to look at the social and disease dynamics in the model without any evolution. And here on the x-axis, we have avoidance. And the level of avoidance is the proportion of contacts with infected hosts that the healthy hosts are not taking, that they're avoiding. And what you can see is this blue line, which is the proportion of hosts that are engaged in social interactions, declines as avoidance of the sick individuals gets higher. And as a result, this red line, the prevalence of the disease, declines. So the avoidance is effectively reducing the prevalence of the disease. And that is accompanied by this gray dotted line, which represents the R naught, or the basic reproductive number of the pathogen. And you can see that as avoidance gets higher, that R naught value falls lower than 1, and the prevalence of the disease drops to 0. And then these healthy hosts can resume their regular social interactions because there are no more diseased hosts for them to avoid. And so with specific avoidance, we've modeled that we can have full extirpation of the disease and preservation of social structure because the healthy individuals are accurately identifying and avoiding infected individuals. Next, if we look at the evolution of resistance through specific avoidance, and so we start with a population that has both the resistant and susceptible types, we can look again at avoidance here on the x-axis is the same as the previous graph. And then on the y-axis, we have the birth rate with a cost of resistance subtracted from that. So at 1 is no cost of resistance to the resistant genotype. And as you go down the y-axis, you have increasing cost of resistance. And then the areas of this graph represent at those combinations of parameters, what happens at evolutionary equilibrium. And so you can see when the cost of resistance is low, resistance evolves. And that is not surprising. Resistance is beneficial, especially when it's not costly. When costs are higher, the susceptible genotype evolves. And then we also have this case where mixed genetic strategies are the equilibrium state. So there's a genetic polymorphism in the equilibrium population. Both resistant and susceptible types are coexisting here stably. And so, and what we see is that this genetic polymorphism happens when avoidance is at high levels and across a wide range of costs. And this pattern is exactly consistent with previous research on physiological resistance. So what we're seeing here is that under particular assumptions, behavioral and physiological resistance can evolve following the exact same patterns. When we look at general avoidance, so this is the case here where avoidance is a reduction of all contacts, not just contacts with infected individuals. You can see that as avoidance increases, the proportion of individuals that are in social interactions declines, along with correspondingly the prevalence of the disease and the R naught of the pathogen. And so these things all decline together, but it's only at the absolute highest level of avoidance that this strategy is effective in reducing the R naught below 1, reducing the prevalence to 0. And when that happens, the social structure of the hosts is severely compromised. Correspondingly, when we look at the evolution of resistance through general avoidance, we see a similar graph, except that it's a lot harder to evolve resistance under general avoidance than under specific avoidance. So these graphs that I've shown you assume a fixed cost of resistance, meaning that the gene that influences avoidance also has a detrimental effect. So for example, individuals that avoid contacting others might meet fewer mates and have fewer offspring. But if we change that assumption and instead assume that avoidance behavior has a direct social cost, so in other words, that social interactions themselves are beneficial and not contacting others is the direct cost of resistance, we find that the susceptible genotype always wins, and the resistant can only evolve in the case of negative costs. And so, at least in our very simple model, no stable polymorphism exists under these assumptions. And that's because the benefits of avoiding disease are perfectly counterbalanced by the costs of these lost social interactions. And so this might help explain cases like the banded mongoose, which is a system I've been working on with collaborators at Virginia Tech. The banded mongoose is a highly social, cooperative breeding mammal, and they have a deadly tuberculosis disease that they transmit in social contexts. And despite the fact that they display outward signs of infection, they don't avoid contacting each other. And it could just be that they're so highly dependent on their social groups that avoiding others is too costly, even if it carries the benefit of reduced transmission. So this example highlights our finding that if costs of avoidance are social, we might not expect evolution of behavioral resistance. So in summary, behavioral resistance can follow expectations from physiological resistance theory, depending on the assumptions, but behavioral resistance is likely to follow different patterns, especially if the costs are social in nature. In terms of future directions, the most immediate steps that we see are to look at how epidemic dynamics might change if individuals can learn to avoid disease within the course of their lifetime. We also want to consider how evolution of behavioral and physiological resistance might interact with each other, because when they're present in the same individual, behavioral resistance occurs prior to physiological resistance. And so these two things are likely to interact, and the genes that influence them are likely to interact with each other. We're also interested in modeling larger groups, which would make it possible to have avoidance within and between groups. This becomes mathematically pretty challenging, so we'd be looking for a collaborator who could take on this challenge. And then I'm actively looking for a study system to examine these processes empirically, either in natural settings or in the lab. And so with that, I'd like to invite you to check out our paper in Biology Letters. I've made a PDF available on my website. And I'd like to thank my advisor, Yanis Antonivics, who is a co-advisor on this research. And of course, to acknowledge the NSF and the NIH, which jointly funded the EEID grant that supported this work. And I'll be happy to take your questions in the time I have left.
Thank you very much, Caroline.
Sure.
We have time for for a couple of questions. Again, as suggested, either feel free to put them into the chat or raise your hand and we'll try to identify, and you can feel free to unmute as well, and we can call on you. Let me start just while people, I know there's always this pause in Zoomdom where it takes a minute for people to figure out how to actually touch any of this. As you mentioned, this couldn't be a more timely conversation to be having right now. Do you see anything, and I guess at some level, speaking that you mentioned even the tension that we're facing today with respect, do you see any translation of this actually from what your models are saying right now as to maybe even philosophically into what how we approach something like the pandemic right now?
Yeah, I really appreciate the question because it's exactly the kind of speculation that reviewers don't like in papers. So I would love to talk about applying this to our current situation. I mean, obviously, our model is a huge simplification of the situation that's going on right now, but those areas of polymorphism, those purple areas of the graph, I think are suggesting that a mixed strategy is going to be what works best here, and that, you know, any ways that we can maintain our social structure, any ways that we can continue to feel connected to each other without you know, paying the risk or taking the risk of transmission in person or, you know, in close quarters indoors, are going to be the way forward, and until we have physiological ways of dealing with this virus. So I think it's sort of a hopeful one in some ways that, you know, luckily we have the technology and the resources in most cases to be able to sort of reduce some of the costs and maintain the benefits of social interaction.
Okay, cool. I don't see any other questions being raised right now, and we're right at the edge of time. Oh, Charlie, did you go ahead?
Oh, I was going to ask a question, but not if it's
Please.
going to cut into the next speaker. Do we have time for it, or?
You can come to my breakout room.
Yeah,
No, that's right. We'll have a breakout room.
I'll defer to the timekeeper Meredith, who was shaking her head. So I do want to make sure that we allow time for everyone. So, but thank you very much, Caroline, and again, great job. All right, our next speaker today is Amrita Bhattacharya. Amrita earned her PhD from Indiana University Bloomington, co-advised by Farah Bashi and Kurt Lively. During her PhD, Amrita examined how social interactions among pathogenic bacteria can affect disease evolution, as well as evolution of resistance against antimicrobials using natural isolates of the insect pathogen bacteria Xenar habitus. In fall of 2019, Amrita started a postdoc in the Kennedy lab at Penn State, where she is delving deeper into her interest in pathogen evolution and investigating factors that can predict the evolution of drug resistance using both experimental and literature surveillance approaches. Her talk today is Exceptions to the Rule: Why Does Resistance Evolution Not Undermine Antibiotic Therapy in All Bacterial Infections? So with that, I'll turn the floor over.
Thank you very much. And thank you all for being here today, especially on a day like this. I'm really excited and honored to be here to share some of the work that I've been doing recently with this audience. And I'll be talking about some of this work that I've been doing over the past few months. Essentially, we started this project when access to the lab was limited due to working from home, and now it has taken off to a pretty interesting project, we think. So it's ongoing work, and I'm very excited to get feedback from everybody here today. Okay, so it is obviously not news to anyone in this audience that antibiotic resistance is one of the biggest global health threats that we face today. And despite being so common, the evolution of resistance itself is not surprising. So by killing off or inhibiting the growth of susceptible pathogenic cells, antibiotics can impose strong selection on any resistant bacteria that can then take off and spread through the population and essentially result in resistant pathogenic populations. And yet, not all human bacterial diseases have been undermined by antibiotic resistance evolution. A particularly striking example here is that of the spirochete bacterium Treponema pallidum, which causes syphilis. And syphilis has been treated with penicillin as first-line therapy since 1944. However, even to date, there are no documented cases of clinical penicillin resistance. So examples like this one and others motivated the study, where we wanted to understand in which cases has resistance evolution not undermined antibiotic therapy, and what factors can explain this variation in resistance. So we started by first using a literature survey approach, where we first compiled a list of common human bacterial pathogens and the drugs used to treat them. We used a standard medical microbiology textbook to generate this list, which gave us 183 distinct pathogen by drug class combinations. This spanned 57 distinct human bacterial pathogens and 15 different drug classes. Once we had this list, we wanted to assess the prevalence of resistance for every pathogen drug combination. Simultaneously, we collected data for a host of drug and pathogen traits that could be important in resistance evolution. And finally, we used mixed effects models to analyze which of these factors explain the variation in our data set the best. Now, the trickiest part of this process was actually assessing the prevalence of resistance for each combination. And this was for a few different reasons. The important ones being that it's really difficult to have consistent measurements across all these different pathogens and different drug classes for resistance. And also there is the issue of some pathogens and drugs being much more widely studied than others. So as a result of these challenges, we decided to use two different approaches to get at this problem. And I want to be really upfront that neither of these approaches are perfect, but they have different strengths and weaknesses. So the algorithmic review method is a lot more precise, where we are looking at 79 specific antibiotics within each drug class and trying to finding 3 most recent papers for every pathogen antibiotic combination, following a very stringent set of guidelines. And some of the important ones being, we're only looking at data from the last 5 years, focusing simply on human clinical isolates. But most importantly, we're focusing on resistance data that comes from published guidelines, such as CLSI or UCAST, or objective measurements in case these guidelines are not available. So the data collection for this process is still ongoing. We have about 1,500 lines of data for this, but the analysis is ongoing. So I won't be showing you too much data from this today. Instead, I will focus on the other approach we've used, which is the expert opinion method, where we have the complete data set. The advantage of this method is that with the precise algorithmic review method, cases where that specific data is not available, we're unable to get a measure for resistance. Whereas with the expert opinion method, an expert, in this case me, conducts the literature survey for every pathogen and drug class combination, and focusing on human clinical isolates only characterizes resistance into one of 3 categories. So these resistance is classified either as very rare or none, where hardly any clinical isolates have been reported so far. Wherever they have been reported, resistance prevalence is below 1%, and it's commonly described in the literature as completely sensitive or resistant. And here I have an example, a perfect type case for this, which is tetracycline resistance in Francisella tularensis. Resistance is classified as rare, where resistant isolates have been described, but the prevalence is typically between 1 to 10%. And this is commonly described in the literature as very low or rare resistance. And a good example here is penicillin resistance in Neisseria meningitis. And finally, we categorize resistance as not rare, where resistant isolates are frequently seen, typically at frequencies greater than 10%, and terms such as increasing strain-specific outbreaks cause for concern are commonly used to describe the problem. And for example, macrolide resistance in Streptococcus pneumoniae. So I collected this data for all 183 pathogen drug class combinations. And in the next few slides, I'll show you what the data looks like. So first I'm going to show you, so before looking at the data, the y-axis here is resistance score, which I just described. So 0 is very rare, 1 is rare, 2 not rare. And on the x-axis here, we have all 57 specific pathogens. So what becomes clear when we look at this is there's clearly variation between pathogens in how much resistance is observed or documented, right? And we find a similar pattern when we look at across the 15 different drug classes as well. So when we look at the different 15 drug classes, there's variation in resistance across each of them. So now, demonstrating that there's this variation, now we were interested in examining factors that could help us explain this variation in this data set. So to do that, we used mixed effects models, where we included pathogen name and drug classes as random effects. And I'm not showing you the data here in the interest of time, but this does significantly improve the fit of the model. And then we looked at 12 different factors that could all be important in the evolution of resistance. Some of these factors are more strongly linked or hypothesized to be important, and others more loosely linked. But I will quickly go through each of the factors that we looked at. So we looked at whether or not the pathogen was nosocomial, which means whether or not these infections are hospital acquired. We also looked at whether the pathogen is a zoonotic, causes zoonotic disease, meaning that there are animal reservoirs. We asked if the pathogens can replicate in the environment outside the human patient, so if they have an environmental reservoir, or if they're part of the human microbiome and thereby commensals. We looked at whether the pathogen is naturally competent. Natural competence is the ability of bacteria to take up exogenous DNA from their environment and is therefore hypothesized to facilitate the spread of resistance. We looked at pathogen type, which is gram-positive, gram-negative, or other classifications. Transmission type, which is whether it transmits directly or indirectly. Whether or not there is human-to-human transmission. We looked at how the drug acts. So does the drug kill the cells or simply inhibit the growth of the cells? And finally, we also controlled for a few different factors, which are how long a drug has been approved for use, so how long penicillin or any of these other drugs have been used, and how much research effort there is. So we looked at how many citations there are for each pathogen itself or for each drug to account for how much a drug or pathogen has been studied. And we looked at every possible combination of these 12 factors, which meant we had 4,096 different models, which we then compared for the best fit by looking at their AIC values and chose the model with the lowest AIC. And when we do that, we find that our best fitting model has six of these factors are included in the best fitting model, each of which here is color coded. Now, interestingly, this is the best fitting model with the lowest AIC value. But when we look at some of the competitive models, there are a lot of models that have a difference of only 2 AIC values, which means they're all very close to each other and can explain the data very, very well. The interesting thing, this is why I've color coded each factor here, is you can see I'm only showing you the 3 most competitive models, but there can be differences in the factors. Some factors from the best model might be dropped off, or other factors might become important. So what we wanted to do going forward was try to understand really how much does each factor contribute towards explaining the variation in our data. And we did this in a couple of ways. First, we took the best model itself, and by either removing one factor at a time or adding one factor to the best model, we asked how much does that change the fit of the model? So when we remove a factor, for example, from the best model, if it makes a huge change to the fit of the model, that indicates that's a very important factor, or it's very important in explaining the variation in that model. So I'm going to show you that data over here, but before getting into that, so I have the change in AIC or delta AIC here on the y-axis. And on the x-axis right now, I have all the 6 factors that were in our best model, right? And the way I've arranged them is in decreasing order of the change in AIC. So the factors that show the greatest change in AIC are important in explaining a lot of the variation. And when we do arrange it this way, we find that natural competence seems to explain most of the variation, followed by zoonosis, nosocomial, transmission type, research effort drug, and human transmission. Now, when we also added all of the factors that were not in the best model, one at a time to the best model. And here we were interested in finding the factor that least made the smallest change to the fit of the model, right? Because then those factors are least, the factors that make a greater change are making the model worse fitting. So when we arrange it that way, the order that becomes apparent is research effort pathogen seems important, followed by drug date, how long a drug has been used, whether or not it's an environmental reservoir, how the drug acts, commensal, and pathogen type. So this order, we believe, from our best model is an indication of the relative importance or the relative significance of these ones. When we look, we also looked at the AIC weight of each factor, which is essentially taking the sum of the likelihoods of every model that contains the factor and divide that with the sum of the likelihoods of all the models. And we arrange, when we look at this data, I've arranged all the data in the same order as we saw before, and that divides the ones that were in or not in the model. Again, we see that the same 6 factors seem to be important in pretty much the same order. So naturally competent, followed by zoonosis, nosocomial, and so on. Now, in the interest of time, I'm going to only focus on the first 3 and quickly give you a glimpse of how these factors influence resistance. And sorry, this is just a slide comparing both of the metrics side by side, just to make the point that they're all in a similar order. So when we look at natural competence, we find that, as expected, cases where competence has been documented show higher resistance, and this makes sense because being able to take up DNA can facilitate the spread of resistant genes. When we look at zoonosis, we find that zoonotic pathogens seem to show lower resistance relative to non-zoonotic pathogens. And this also makes sense because a lot of these zoonotic reservoirs happen to be wild animals, for example, deer for Lyme disease, and those do not get exposed to antibiotics outside of when they're being treated in a patient. And finally, unsurprisingly, we find that nosocomial pathogens seem to show greater resistance. And this also makes sense given that in the hospital environments, exposure to antibiotics is pretty high. So just to quickly wrap up, we wanted to understand when is antibiotic resistance less likely to evolve. And based on the data and the information we have so far, we identified a few factors that may be important. And among these, we find that pathogens that are not naturally competent, not nosocomial, and zoonotic are likely to show lower resistance. And we also identified a few other factors that seem to be important in explaining this variation. So we're continuing this work by analyzing the algorithmic review method data and getting an additional expert to also assess resistance prevalence. And with that, I'd like to thank everyone in the Kennedy Lab, especially collaborator Anton and my advisor Dave. And I'm happy to take any questions. And thank you all for the time and attention.
Okay, very good. Thank you. We have time, I think, for a quick question. I see let's see, Charlie has one in the chat. Do you want to ask that, Charlie?
Yeah, sure. I was just wondering what you think about prevalence of the pathogen in the population as predictive of resistance. Just, for example, as a proxy for how many courses of antibiotics have been given or something like that, if you look at that.
Yeah, that is a really good question. So the way we're trying to, so you're asking prevalence, so how common a disease is and therefore how likely it is to be treated. So that is a great question. And we do account for, it's not exactly how common a disease is, but we do account for research effort in pathogens, which is how many citations there are. So we believe that that is one way of getting at how important a pathogen is believed to be or how much it is studied. In terms of, so we're hoping that that accounts for at least part of that kind of variation. But it would be interesting to be, if we were able to get data for antibiotic doses. It's a little bit tricky given that we're looking at global patterns of resistance and specific antibiotic doses could be, but we do look at research for drugs also.
Okay, thank you very much. I think for sake of time, we'll have our break room later that we can actually dive deeper into additional questions, but so that we can have everyone have time, we'll transition to our final speaker of the session. Our final speaker today will be Angela Garcia. Angela is currently a postdoctoral research fellow at the Center for Evolution and Medicine at Arizona State University, and actually a colleague of mine, so just in the spirit of volunteering conflicts. She will be joining the faculty of the Department of Anthropology at Emory University in July of 2021. Her work is situated at the intersection of human biology, evolutionary medicine, and anthropology. Angela uses a life history framework and a multi-level systems biology approach to investigate how social and ecological factors impact endocrine and immune physiology and contribute to variation in health and disease between and within populations. Angela's talk today is Evidence for Height and Immune Function Trade-offs Among Pre-Adolescents in a High Pathogen Population. So with that, Angela, the floor is yours, or the virtual floor is yours.
Thank you. Thanks, Ken. Thank you all for being here. This is definitely a surreal time, I think, for everyone. So I appreciate that can be a distraction maybe for a few minutes. Let me share the screen real fast. Okay. And all right, I think that should be good. Oops, maybe not. 1 second. Is my screen shared?
It is not yet.
Thank you. I'll try this again. One would think I would have gotten this right out. All right, 1 second. Now I believe it is, right?
Yes, but.
Better?
So yeah, there we go.
Okay. All right. So thank you all. As Ken said, I'm going to be actually talking about a paper that we published a couple months ago in the EMPH journal. So to kind of situate us, energy available to organisms is limited and is allocated to competing physiological demands in ways that optimize biological fitness. So given resource limitations or constraints, it might necessitate trade-offs between key life history features, things like growth, reproduction, or maintenance. And specifically, I'm interested in immune function. We're talking about maintenance. Now, a trade-off is any situation where increased investment in one biological function will result in a decreased investment in a different biological function. Natural selection can produce adaptations for managing these trade-offs in species typical ways, as in the case of the anglerfish here, where the male essentially attaches to the female permanently during mating. So they, as a species, they essentially traded their adaptive immune system and for the male's growth to increase greater potential for reproductive success. But natural selection can also select for norms of reaction that manage allocations to fit specific ecological contexts through developmental plasticity, which allows individuals to essentially reach their optimal energetic strategy for a particular environment. This is some work by Juan Solar and others that found that magpie nestlings that were more heavily parasitized or showed greater immune response were actually smaller in size. Now, the force of natural selection declines as a function of age. And it's really in these earliest years where the human body is calibrating allocations across competing demands, specifically between growth and immune function, when natural selection is really operating the heaviest. And it's given this strong selective pressure against early life mortality, early investment in immune function should be prioritized despite competing energetic requirements like growth. And you can think about this like there's always the opportunity for catch-up growth. But if you don't invest more in immune function, you might not make it to that opportunity, right? And this is particularly salient in high pathogen environments where investment in the adaptive arm of the immune system, which confers long-lasting and specific immunity, would be critical for surviving that early period. Now, growth and immune trade-offs might represent short-term accommodations to temporary research shortages or longer-term phenotypic adjustments to expected future conditions. So when we're thinking about short-term accommodations, there's actually been some great previous work done in the Shuar, which is another population from Ecuador that are quite high, they have quite high pathogen prevalence. And Sam Erlacher and colleagues found that we do actually see, or they do actually see a trade-off between CRP and growth. So CRP is a marker of acute immune activation. Kids that had higher CRP grew less, but this was moderated by body fat. So kids that had more body fat saw less of a cost on growth. And this makes sense in the short term because fat stores represent this critical energy reserve that your body can draw on to fuel these short-term metabolic costs of infection and growth. But this is over only a week of time. And we don't know a lot in humans about what this looks like over the longer time period. Now, for longer term adjustments, thinking about anticipating future exposures, we might see a more permanent trade-off due to heavier overall investment in immune function, which cuts out growth. So my hypothesis going into this was that in a high pathogen environment, it should favor early investment in adaptive immune function at the cost of somatic growth. And I am particularly focusing on adaptive immune function because the costs required for adaptive immunity are heaviest during the developmental period, during the early developmental period, because your body is investing resources in the development of things like the thymus and the pool of naive cells. So given the concurrent costs in that narrow window with growth, you should see trade-offs here. And the questions I was interested in are, one, are growth and immune trade-offs mainly due to past exposures, so responsive or short-term, or in anticipation of future threats, so this predictive or longer-term adjustments. And do energetic resources moderate heightened immune trade-offs? So in this paper, we used flow cytometry to examine both innate and adaptive markers of immune function. And one way to address the kind of question of short-term accommodations versus long-term adjustments and trade-offs is to look at cells that both represent immunological resources that have not yet been activated, so looking at the naive pool of cells, and cells that are indicative of past exposures, so non-naive or memory cells. This work was conducted among Chimane children of the lowlands of Bolivia from 2 months to 8 years old. The Chimane are foraging horticulturalists who live in the lowlands of Bolivia, as I just said. They live primarily along the Beni River Basin. They're relatively calorically limited. They practice slash and burn agriculture and hunt and fish, which constitutes most of their calories. They also face quite high pathogen exposure to a wide range of different pathogens. So for instance, for the kids in this study, about 60% of them had helminthic parasites and about 30% had protozoal parasites, things like Giardia. Past work in the population done actually by co-authors has found that the Chimane invests overall heavily in immune function compared to US and European populations. You also see a more rapid depletion of naive adaptive cells, which suggests high pathogen diversity. And we also know that the Chimane are quite short comparatively. And this paper actually uses Chimane-specific z-scores that are derived from these population-specific growth curves. So I asked, is there evidence of heightened immune trade-offs among Chimane children? Now first, this is just looking at the changes in immune cells over time. And so we do see a decrease over age for all adaptive immune cells and an increase actually in natural killer cells. Now next, I ran a series of mixed effects linear models that tested height for age as a function of different immune cell counts. And you can kind of focus in this box. This is where all the adaptive immune cells, cell counts are. And what I found was that consistently you see this negative association between both naive and non-naive cells. So essentially, taller kids have lower levels, taller kids for their age have lower levels of these immune cell counts, or the opposite, right? Higher immune cell counts means that they're shorter. Oh, and just quickly, that also, if you adjust for parasitic infection, that gets stronger. All those associations get stronger. Now, because children traverse through numerous developmental phases, and especially at the early ages, before the age of 3 and before you stop really breastfeeding, infants' energetic resources are still really quite tied up with their moms, right? So we also examined associations between height for age z-scores and immune cell counts that include an interaction term for age groups. So under the age of 3, so 2 months to 3 years, and then from 3 years to 8 years. And what I found was that among infants, there are actually no associations, no significant associations between height for age and immune function. So I wanted to then evaluate whether maternal or child energetic status moderates the associations between height and immune function. So I looked at an interaction between energetic status and cell counts in predicting height for age. And here, what I found was that for kids whose moms are energetically buffered or have sufficient energy stores, these kids actually have a positive association between their immune investment in immune function and height. And sorry, when I say kids, I mean infants, so under 3, right? You do not see that for moms that have below the mean BMI. You still see that negative association. And as you'd expect, when moms are no longer directly providing energetic resources for their kids, you don't see an effect of maternal BMI on that, those associations in pre-adolescence. Now, looking at child energetic status, for infants, we don't see any relationship or any modification as a function of the child energetic status. But in pre-adolescence, so over the age of 3, what we find is that kids that are above average in weight for height, Z-scores, which is what I use as a marker of child energetic status, you actually see greater trade-offs between immunity and height for age. So it's in the opposite direction of what we expected, because the interactions are negative. And to dig into that a little bit, I then compared high and low weight for height pre-adolescence. And what is going on is that for the high weight for height group, they're taller for their age than the lower weight for height, but they don't differ across any parameters of immune function. And we're going to talk about this, or I'm going to talk about this in just a second. To quickly summarize these findings, though, I did find partial support for the hypothesis that in a high pathogen environment, it should favor early investment in adaptive immune function, and you should see a trade-off against growth. Because we find these consistent negative associations between height for age and both naive and non-naive adaptive immune cells, which is strengthened when adjusting for parasites. For infants, maternal energetic status appears to buffer those trade-offs, but for pre-adolescents, child energetic status moderates it in the opposite direction than expected. And as I just showed, higher weight for height kids are taller, but don't differ in adaptive immune function. So there is some evidence for these growth and immune trade-offs, and we conclude that it's not strictly due to past exposures. And I want to kind of unpack this a little bit. So you have naive T cells, and these are cells that have not yet encountered a pathogen or an antigen, right? So reduced cell counts might represent past exposures if exposure converts naive to non-naive cells, right? However, if that were the case, then you should see a positive association between naive T cells and height, but we don't see that. In contrast, if higher naive T cells are representing investments in immunological defenses for threats that haven't yet been encountered, then you should see a trade-off with height. And we do find that. And the fact that both cell types, both naive and non-naive cells, are negatively associated with height, even when you adjust for current parasitic infection, this suggests that trade-offs with height are likely due to an overall investment in cellular immunity, not necessarily dependent on the causes for that investment. So it doesn't, not dependent on whether it's responsive or predictive. And these associations also suggest that it's immune investment itself that may trade off with height rather than just the direct cost of past infection. Now, to understand a bit more about what's behind this lack of heightened immune association in infants, well, so some past work with the Chimane, Melanie Martin found that Chimane kids breastfeed until about the age of 3, which is why I chose that as a cutoff. And it could be that infants are able to maintain adequate buffering by maternal energetic and immunological resources transmitted via breastfeeding. And in accordance with this, we did find that positive interaction between mom's energetic status and kids' investment strategy, or babies and infants' investment strategies. And then finally, what's going on with this negative heightened immune association being higher for higher weight for height kids at the proximal level. This is explained by this difference in height and no difference in immune function. But the fact that these groups have similar adaptive profiles suggests that immune function must be maintained in the face of energetic constraints. It's likely that in a high pathogen environment, there's greater plasticity in height, whereas there might be this sort of cliff edge effect for immune function, such that you have to invest sufficiently in adaptive immune function, because if you don't, it's literally deadly. And it's critical for survival. And any additional resources might be invested in height, but not at the cost of meeting that baseline of immune function. But also, over-investing could also be problematic because it puts you at risk for things like runaway immune responses or things like sepsis or autoimmunity. And we obviously cannot conclude this from this data, but I think it speaks to an interesting question in the immunological literature of this notion of bounded optimalities that are found in things like MHC diversity and cliff edge effects. And really, we know quite little about adaptive variation in immune function in humans. And these findings suggest that we really need more research to investigate the costs and benefits of immune variation across different ecologies. And with that, I realize I'm running short on time. Thanks to Chimane folks and my mentors and a lot of great reviewers and people along the way. Thank you. And all right.
Thank you very much, Angela. I think we have time for a couple of questions here. Does anyone, again, raise your hand or enter into the chat box? Anyone have anything they? So we have one question in the chat that says, benefits of height, it's from Bernie Crespi, benefits of height ought to be affected by sex. Does sex affect the trade-offs?
That's a good question. So let me think about this. I didn't explicitly test interactive effects with sex. I just adjusted for it. So, and that would be an interesting thing to go back and look at. And I'm thinking about, because I use the height for age Z-scores, even within the calculation of those Z-scores, they account for sex variation. But I mean, that's a great point. And
do you have a prediction about that at all? Or, I mean, if males might follow a more risky strategy, perhaps?
Well, right, but then I guess I would still push for this notion that I would see that maybe being the sex differences coming into play more when we're looking at how energetic status plays into that, because I do still think that regardless of the sex, there's going to be this kind of threshold of having to invest sufficiently in immune responses regardless of your sex, right? And so it might just be that males see greater investment in height if they are able to afford it. But otherwise, I guess I think it'd be more extreme maybe for the height trade-off with males than females potentially, but I don't think it would impact the immune investment necessarily.
Thank you.